Facial recognition is an evolutionary process that played an important role in social encounters for millions of years by providing details about the physical and psychological states of individuals in both human and non-human species. The literature on face processing describes the existence of multiple models of face recognition, such as Bruce and Young in 1986 or Spector and colleagues in 2017. Duchenne and Yovel's 2015 one, however, stands out by proposing an independent dual-stream processing of faces model that involves a dorsal stream focused on facial expression recognition, and a ventral one that is focused on the processing of personal identity features. Only one brain structure appears to be interconnected during the early processing of a face, the posterior superior temporal sulcus, therefore suggesting that both personal identity and emotional expressions share some common processing similarities. Several event-related potentials have been associated with various steps of successful facial recognition, and the ones that we are primarily interested in are represented by the P1, which accounts for the processing of the visual characteristics of a stimulus, the N170, which is the potential associated with the cortical distinction between a face and a non-face, the N250, which is associated with a prior familiarity with a face, and the P300, which accounts for the processing of the emotional expression. Most of these ERPs can, however, be modulated in time course and amplitude by providing participants with unknown and difficult to recognize stimuli to observe whether any task-induced events occur there. In our experiment, we wanted to find out whether emotional expressions play an important role in the short-term facial recognition of personal identity features. Therefore, we have opted for a mixed behavioral and electrophysiological approach to measure the reaction times and accuracy of recognition, as well as to observe whether these differences can be observed in the time course and source localization of the event-related potentials. For this, we have used a 3x3 design, where our independent variables were emotion, with happy, neutral, and angry conditions, and level of morphing with low, medium, and high conditions. The experimental structure was composed of a structural encoding stage, where the participants were familiarized with 36 homogeneous identities, which was followed by a behavioral task where the participants were presented with a familiar face used as a prime, and an unfamiliar one, which accounted for one of the three before mentioned levels of morphing, and they had to provide similarity ratings for the two faces. The experiment then ended with a repetition of the first task to observe whether there are any effects of short-term familiarity with the faces. Preliminary results reported after the analysis of 26 out of 53 datasets showed that there was an effect of emotion on mean similarity ratings, with the neutral and happy faces displaying the highest scores, whereas the similarity ratings followed a gradual decline with increased levels of morphing. Further analysis is required to observe how the ERPs associated with the processing of a face interact with these similarity ratings from a mixed behavioral and electrophysiological approach, where we would expect higher ERP amplitudes to correlate with greater similarity ratings and faster reaction times.